Howdy everyone, my name is Jiwan. So we have our second last keynote speaker. It's my honor and pleasure to introduce one of our keynote speaker and my PhD advisor, Dr. Isabel Vales. So Dr. Isabel did her master's and PhD from Spain. So after doing her PhD from Spain in maize genetics, she went to University of Minnesota as a postdoc. After doing postdoc, she transitioned to Oregon State University as an assistant professor. From there, she got chance to lead the potato breeding program at Oregon. She has quite a bit of experience working on different aspects of the legume breeding as well. She was a principal scientist at ICRISAT India for a few years. Then after that, she has also experience at Washington State. She has released 18 different potato varieties up to now. So without further delay, I would like to welcome Dr. Isabel Vales.
Thank you very much, Yuan, and thank you for, to the committee members organizing this symposium, and also to our sponsors, Corteva and the other companies. Thanks to everybody for coming to listen to my talk. I wish I could talk about coffee because I feel a little sleepy myself, but no, we are going to talk about potatoes. This. All right, so I will share with you information about the Texas A&M potato breeding program and my efforts to transition from a conventional breeding program to a molecular breeding program. The outline of my talk will include an overview of the program as well as examples of some of the Texas A&M potato releases. Then I'll continue talking about challenges in potato breeding, and I'll share with you some of the highlights of my program right now, including studying the genetic diversity of the breeding pool I have, marker-assisted selection, genome-wide association studies, heat tolerance, zebra chip tolerance, and then I'll touch some new areas of exploration, including studying the chemical composition of the potatoes, leaning towards health and nutrition, precise phenotyping or alternative ways of phenotyping, some gene editing collaborations, and genomic selection. I will not talk much about genomic selection, but I'm a believer, okay? It's just that in the process of moving from my conventional breeding program to the from my conventional to the molecular, I have to go stepwise. But I have to share with you that we are working on a proposal together with Oscar and Dave and others, CRI polyploids. So I would like to, go along and try to implement some of those technologies. All right, before moving into the potato program, just some quick facts about potatoes. Potato is a third most important human food crop and definitely the #1 vegetable. It was domesticated 8,000 to 10,000 years ago around the Titicaca Lake, southern part of Peru, northern part of Bolivia, and now it's grown in more than 160 countries. That's really unbelievable. It's definitely the leading crop in terms of production of energy per unit area. And we defend it as a really healthy crop. It all depends on how you eat it, but if you don't use lots of oil and salt, it's good for you. And precisely because of that, high levels of vitamins and minerals, fiber, and other phytochemicals, together with the fact that we eat lots of potatoes. More than 110 pounds per year, even small changes in the chemical composition could have a huge effect on our health. Now, I'm going to talk a little bit about potato market classes, and I wish I was at the point of telling you that we sell potato by variety name, but we are not that sophisticated yet. In Europe, they are doing a much better job with that, and I just hope that the US follows the trend that we see, for example, in apples. In apples, they have done a tremendous job, and most of you probably have your favorite apple based on the level of acidity and sweetness. In potatoes, we pretty much talk about two major market classes, which are the processing on one hand, and the other one is the fresh market. The processing potatoes are the ones that are used to make French fries, potato chips, and dehydrated. And for the fresh market, the way you probably refer to the potatoes are mainly based on the type of skin. So the ones with that netted skin are russets. And then we have the yellows with yellow skin and the reds, and that's pretty much it. We don't go by variety. But the market is evolving, is improving. So there are some emerging new types that are specialties, really small potatoes that come in containers that are very convenient because you put in the microwave and you get your dinner pretty much right away. So there's room for exploration there. Now, in terms of the Texas A&M potato breeding program, I have to let you know that the program was established more than 40 years ago by Dr. Miller. And we are really sad that he passed away last November. The goal of the program, as it was stated by him, was to develop new potato varieties adapted to Texas that result in economic profits to the industry and superior products for consumers. I'm building up on that foundation. He had a really good program, and pretty much my goal is not going to deviate much from what he has already established. What I would do is like redefine some of the aspects. So when I think about improved, I think about high yield and high quality. But now, because of the higher demands of society, we have to also think about healthier, more nutritious, and environmentally friendlier. When I think about environmentally friendlier, I think about potatoes that have disease or pest resistance, host resistance. So those potatoes can defend themselves against the threats. But not only that, also that potatoes tolerate the environment, because specifically in Texas, very high temperatures, potatoes have to grow under those conditions. So potatoes need to also deal with those abiotic stresses. And then adapted to Texas, for sure, and we have to continue working on that. But I now know by fact that potato varieties released by the potato program in Texas are doing pretty well nationwide. And not only that, even internationally. And so if they perform well here, it seems like they perform well in many other places. We are really hard and tough at selecting them. And then profits for the industry, but also to the profit, you know, we want to see benefit for the environment as well. And that's a little more difficult to measure, but if we can start using less pesticides and think more about fertilizers and improve the quality of water, that's on the best for the industry. And consumers, but I want consumers with capital letters, because they have a lot to say in terms of the type of products they want. So we need to be very conscious about that. And then leave a door or a window open for exploration, for new trades, new application, because maybe the consumers are not demanding it, but we could run into something that is really good for them. It seems like many of us maybe were not demanding cell phones, but now that cell phones are available, everybody uses cell phones. So maybe we have to be the ones saying, this is what we have. Do you want it? Try it. Do you like it? Essential to the potato program is the people. And I'm going to introduce my core team. Definitely still want to keep Dr. Miller right next to me, because I will be really building up on what he has constructed over so many years. Then we have my right and left hands, my senior research associates, Doug and Jeff, and as well as some other assistants. Angel, she moved to another area, but I really appreciated all she has done for me, and Maitri, and Mike Jensen assisting Jeff in Lubbock. And then as for graduate students, Sam worked on the zebra chip project. He finished his master's and is now working for private sector. And some other students are right here. We have G1. And G1 has been leading the genetic diversity, GWAS, and cold switching studies. And we have Sanjeev as well, and he's been working on heat tolerance as well as dormancy. Tanya Sharma, she's done some of the marker work for some of the major genes. And then we have many undergraduate students that are really essential because there's a lot of labor involved in potato breeding. Now, it's not only in Texas. I actually have cooperators all over the US. Within Texas, in addition to the breeding program, I depend on pathologists, entomologists, food scientists. Then the potato program of Texas is really well integrated at the southwestern level. So it's Texas, Colorado, and California. And the next level up includes the Western Regional, those 3 states, plus Idaho, Oregon, and Washington. And going even farther with many of the national programs, especially related with all of the chipping work. And the USDA ARS, as well as the Potato Gene Bank that provides new genes for the program, and they've been really helpful. The other part that I am very thankful for is my cooperators, is the growers. My field trials are in growers' fields, and they have provided a lot of in-kind support for the program. Facilities, in College Station, I have greenhouses, and as well in Lubbock, that's where we mainly do all of the crossing and the development of the families. As we have tissue culture facilities where we maintain all of our stocks, as well as a basic molecular biology lab and the dry lab. And then a field where we do our field trials, storage, both in Lubbock and College Station. And in terms of processing, the main processing we do is related with making potato chips. Texas is a big state. I came to realize that. So my field trials are in Dalhart, all the way to the north, Spring Lake and Weslaco. And then we have two kind of core centers. One is College Station and the other one is Lubbock. Like you can see in those pictures, you see lots of circles. I mentioned earlier to you that my field trials are in the middle of potato production, and I am using land that belongs to the farmers. In the case of Spring Lake, they just rotate us around, but in Dalhart, we have a designated circle, which is maybe a little difficult to see, but it's like a little circle in the middle of the large circles. And I don't know if you see my pointer, but it's like, do they see the pointer? No. So it's a little circle of 35 acres in the middle of the large ones. You see now? Oh, I have to point out here. You see that little circle in the middle of those large ones? That's 35 acres. 35 acres. I use 1/4 each year and keep rotating. But that's for us, all ours. So that's really good. It takes 8 hours to go to Spring Lake driving. It takes 12 hours to go to Dalhart driving. We always stop at Buc-ee's. And in the last 2 years, I also had trials in Weslaco, which is another, 6 to 7 hours south. So if you think about planting and evaluating and field days and harvesting, and we really interact a lot. We really know each other. We have lots of time to talk and share ideas. The main objectives of the program are to develop high yielding and high quality potato varieties. And the main market classes that we are focused on are the processing chippers. It seems like high temperatures in Texas are giving us a hard time, so we cannot really produce very good French fried russets, but the chippers are doing pretty well. Just recently, we ran into some processing russets with very high specific gravity, but that's not the norm in Texas. And in terms of the fresh market, it's mainly the russets that you buy from the supermarket for baking or microwaving, then the red skin with white flesh. And I've been working quite a bit in the specialties as well, those with different sizes and shapes and colors of the skin and flesh. There is market for that. The markets are actually developing, but we think it's gonna be a reality very soon. In terms of priorities, I'm listing here some of the research areas. I listed them in the order that I think they are right now, but that was not the case when I started the program because zebra chip was the #1. Now it's going lower in that scale because our growers are doing a really good job controlling the disease. But in any ways, I worked a little bit on that as well. So PVY, potato virus Y, is very important, especially for seed producers because the allowances for the viruses are very low. And since potatoes are vegetatively reproduced, they keep accumulating viruses and bacteria over time. So having this resistance is really critical. As well as late blight, more and more as our growers want to go into organic production, and then nematode resistance. Now, high temperature tolerance is a wish in Texas, definitely, and that is quite often linked to earliness. Dormancy as well, the time it takes from when we harvest all the way until the tubers start breaking that dormancy and germinating is really critical to extend the time we can preserve potatoes. Cold sweetening resistance is also related to storage, but more in the case of the chippers. When we store chippers in cold storage, there is accumulation of sugars, and then those sugars will, when we fry them and chip, they will combine with free amino acids and give you a brown product that is highly undesirable. But we have some materials that can go into cold storage and you chip them and still give you light chips. So that's really good, working on that. I'll show you some information about the zebra chip efforts in terms of looking for sources of resistance and tolerance, and some health and nutrition, which is an area that I think is gonna grow more and more in the near future. In terms of strategies, I'm really paying a lot of attention to parent building, bring materials from other programs that carry genes of interest. Since the cultivated potatoes are tetraploid, I want to have parents that have multiple dosages of the desired genes, so that way when I use them as parents, I have much better chances of obtaining progeny with the desired combination, as well as pyramiding of major genes and eventually quantitative genes, as well as recurrent selection. In terms of parental building, I think it's going to become really critical, the genomic selection, because if I start bringing into the crossing blocks parents with very good breeding values, my chances of developing excellent progenies will increase dramatically. Another thing that I've done is develop breeding data management system, because the program had many years of data, but everything was kind of dispersed in Excel files. And so we have already compiled more than 10 years of data, which is going to be a really great source of information for me, for the program. Very critical as well is experimental designs to take advantage of early generation and use augmented. So we start using more of the statistical information down the line. Precise phenotyping, I cannot emphasize that enough. The quality of the results is only as good as the quality of the phenotype and the genotype that you do. And as well as implementation of a statistical analysis that include mixed models that would allow us to account more for the variation. Market assisted selection for major genes, there are a few, not many, but there are a few. Genome-wide association analysis, genetic mapping, QTL analysis, that's kind of connected with gene discovery, gene editing, and genomic selection. Now, in particular about the approach on how we go about developing the material, everything starts by selecting the parents, and once we select the parents, we cross them, and this is the type of fruits that potatoes have. Those are berries that have seed, very tiny seed, true potato seed. The true potato seed is planted, and we get full seed families that segregate for the traits that were present in the parents. Initially, selection is phenotypic, and at just single hill level, or later on, as we keep increasing more and more the material vegetatively propagated, we have enough for replicated trials at many locations where we can start looking at agronomic parameters, quality parameters, disease, pest resistance. Since in Texas we don't have a potato certification program, it's really essential that the moment we have promising material, we bring it into the system, and we have to clean them up, free them up from viruses. So we conduct ELISA and test for the presence of 7 viruses. If they are not clean, we have to do chemotherapy and thermotherapy, and then they enter the system, and we start propagating them to be able to move them to regional trials, and then down the line all the way to commercial trials and protecting the material down the line. This scheme is pretty complex, and I'm just going to point out a few things. It's the scheme of my breeding program. Everything starts selecting the parents and crossing. In green are the steps that take place at the greenhouse level. So we have 2 years in the greenhouse, the first year making the crosses, the second year producing the full seed families. And my crossing block typically has around 80 parents. The full seed families, we end up sharing some of them with other programs, and we receive some from other programs. At single hill level, which is the first field level, so that's in brown would be our field years. The first field year is what I call the single hills. Last year I had 70,000 different genotypes under this step. And I select pretty harshly. So out of there, only like 1% move to what we call the 4 hills. Then from there, they go to Texas level trials, which is 1 or 2 years. And I said that I started cleaning and then moving into regional trials, first at the Southwest level, so it's Texas, Colorado, and California, and then to another level of regional trials that in addition to those 3, we have Idaho, Washington, and Oregon. And in parallel, we have the national chipping trials right here. So down the line, we start with many of them, and we end up with just a few that will go into commercial trials, plant variety protection, and hopefully one of those will be a variety. I'm going to show you some examples of potato varieties that have been developed by the program over time. The first one is actually a series of clones, of potato clones, that are clonal selections of Russet norcota. After being evaluated with many markers, the differences are really very difficult to identify. So it's probably epigenetic variation, and they will need to be studied in more depth. Looking at the historical data sets, I noticed that in regular years or good years, there is no difference between the clonal selections and the original Russet norcota. You really start seeing the advantage of the clonal selections if the years are pretty challenging, like it is really hot. The second one is Sierra Gold, a potato with that netted russet type skin and yellow flesh. It was released exclusively to Calor in California. Next is a red potato with yellow flesh, also exclusively released to Calor. Reveille Russet was named after our mascot, the Reveille dog, and the PVP was submitted in 2015, and just last year we submitted it for plant registration in Canada. There are 18 licenses, and the number of people interested in this potato is increasing pretty fast. It has good shape, good for the fresh market, long dormancy, is heat tolerant, and so we have lots of hopes in this variety. Another one that the PVP was submitted in 2018, and it was submitted for registration in Canada last year as well, is Vanguard Russet. And one of the key features of this potato is the shape more than anything, is the looks. It just looks really good. It's a blocky potato and it's heat tolerant. Pretty much all of the ones that come out of Texas should have that, otherwise they don't make it. And another one that is pretty unique, and I don't know if you can see it from where you are, but it's a russet potato with pink eyes and yellow flesh. And I'm currently negotiating the final steps of an exclusive release. This is one of the few that actually has high specific gravity, high level of starch, even under our conditions in Texas. This next one is kind of like a novelty, and we are exploring commercial potential. It's a bicolored potato. It's a little flat, which initially I thought it was a disadvantage, and then suddenly somebody said, oh, we really like it because it doesn't roll down the chain, so it doesn't get bruised. You never know, sometimes those things are good. Another one that is pretty interesting, like kind of like a fingerling, red skin, red flesh, has very high levels of antioxidants, and I think it would do really well for vegetarians, so they add a little bit of color to their plates. There are many more in the pipeline, so you can see that just talking about the general market classes based on processing fresh might not be the case in potatoes. Potatoes could definitely be sold by variety name, in, you know, like in the case of apples, and the more the market gets sophisticated, maybe there are like specific potatoes for specific dishes. So we all will have that information hopefully in the near future. So in parts of the potato program, the more than 17 varieties have been released over time, and the TAMU releases represent 8% of the seed planted in the US. The program has brought a significant amount of royalties to the university, and some of them are reinvested in the program. Most of the russet type clones in Texas are the russet norcotas at this point. Potato production in Texas is not large, maybe around 20,000 acres, but it has really high value for the growers because they can provide fresh market potatoes when other states are not ready yet. So the price our growers get is $15.2 per hundredweight versus like $5 in Idaho, let's say. So potatoes really contribute to the economy of Texas very well. Now, as we think about challenging in potato breeding, most of the cultivated potatoes are tetraploid. So they are autotetraploid. Basic chromosome number is 12, and there are 4 copies for each chromosome. Total number of chromosomes, 48. It's highly heterozygotes. So for a particular locus, you could have up to 4 different alleles. And the segregation is really complex. It has tetrasomic inheritance. And clonal propagation is also a problem or an advantage, let's say. Once we have the genetics fixed, we can just reproduce vegetatively, which is great. But clonal propagation comes together with accumulation of diseases and the need of going through tissue culture. Linkage drag, as we cross with wild relatives, we could bring lots of undesirable traits. There is cross incompatibilities, self-incompatibilities that make our life really difficult because we could plan a cross and it doesn't happen. There is, even in the case that they are self-compatible, there is inbreeding depression, which is really undesirable. And then we have the issue of tuberization response. The potatoes from South America are adapted to produce tubers under short days, but here in the Northern Hemisphere, we want them to produce under long days. So bringing materials from different places as parents could bring some of these complications. But the good thing is that we have lots of genetic and genomic resources. In terms of genetic resources, there are thousands of varieties available that could be used as parents, as well as land races, primitive cultivated species. wild relatives that carry very important disease pest-resistant genes. There are mutants, cytogenetic stocks, and very good germplasm banks that are helpful in terms of providing materials whenever we need them. In terms of genomics, we have cDNA libraries, genomic libraries, expression profiles. The potato was sequenced in 2011, and since then, others also joined, like the diploid M6. And right now, there is an international consortium, a Pangenome consortium, to sequence cultivated varieties. And there are 4 varieties from Europe and 2 from the US that are being sequenced. We want to understand more about the haplotypes and so on. Texas A&M is a part of this international consortium. And then many other things, including markers. For markers, the ones that I'm mainly working on right now are SNPs. And there are different arrays. We already went from version 1, 2, 3, and 4. Version 1 had 8,000 SNPs. And the one I'm using is version 3 with 22,000 SNPs. I'm not sure how many version 4 has, but it's about to come out. There are diversity panels, mapping populations both at the diploid and the tetraploid level. For many of them, there is genotypic information and phenotypic information. In terms of software, lots of advances. There is software to do genetic mapping and marker trait associations at the diploid level, but more and more are being developed at the tetraploid level, and we are very excited about it. I've been using, together with my students, a GWAS Poly developed by Jeff Enderman in Wisconsin. So genetic maps, major genes and QTLs, databases. We can also benefit from other crops that belong to the same family, to the Solanaceae family, like peppers, tomatoes. There are transformation platforms in place, work done by the private sector specialists in plotting the introgenics and cisgenics, gene editing, and the important part, I guess, is the translating a lot of the basic research that has been done to more of the applied avenues. So we can start using marker-assisted selection, genomic selection, and so on. So now I'm going to give you a little bit of an idea about some of the things that we are doing in the program, kind of moving towards molecular breeding. And I'm building all of this on the material that I had in the clone bank. I had a really good clone bank that pretty much represents the breeding pool. And that has happened because there was this need of bringing fast and cleaning fast some of the materials. So I have more than 200 clones that include different types. And those clones have been propagated in the lab first, then in the greenhouse, and they were tested under field conditions for a number of traits, more than 20 traits. And like you can see in that picture, there's a lot of diversity, lots of things that we could be exploring. So the first thing was to look at the genetic diversity of that pool, and the version 3 Infinium SNP array was used. And we looked at the structure and many other things, pretty much dividing the material into 3 main groups, russet, the chippers, and specialties. That effort was also really good to identify just a few duplications or inconsistencies with the names. And all of that has helped us to define unique fingerprints that we are using for protecting the varieties, selecting parents, and removing duplicates. Some marker-assisted selection is taking place for major genes, and we don't have many, but a few: resistance to PBY, lay blight, Columbia root non-nematode, and golden cyst nematode. And the intent is screen parents, screen all of the diversity panel that we have, and then start applying marker-assisted selection after the single hill stage in the field. And that will guide us in terms of which clones we should maintain, keep. A few examples of our effort in in genome-wide association studies. Like I said, many traits have been evaluated. Here you have some of the Manhattan plots for russet skin, the level of russeting in potatoes, as examples for the length width, which pretty much represents they go from round to very long. Dormancy, which is also very critical because the more dormant the tubers are, the better we can store them long term. Some factors have been identified. The whole diversity panel will be screened for heat tolerance as well. And these are some of the typical defects that you can see when potatoes are exposed to heat. So you have heat sprouts, chain tubers, knobs, internal heat necrosis, pretty terrible. The first thing that we wanted to know is, are our locations good to screen for heat tolerance? And the answer was yes, both of them are. A selection of a planting date. We checked a couple of planting dates, but we are going to stick to the regular planting date because it gives those type of defects, so that's good. And then identify susceptible and tolerant checks that we can use for other studies. And right here, we have information about total yield of US #1s out of our 2 locations, Dalhart and Spring Lake. Yield in Dalhart is much higher than in Spring Lake. And then when we look at percent calls will represent the percentage of external defects, we can really tell that Russell Burbank, their #1 processing potato in the whole US, doesn't like Texas at all, very high percentage of calls. And then there is an Atlantic, which is a cheaper variety, #1 chipping variety in the US, and is terrible for internal defects. You still see high yields, but really lots of internal defects. So I know now that Russell Burbank and Atlantic can both be used as susceptible checks. On the other hand, some of the Texas materials are doing pretty well. And for example, you have here like the Russet, the Reveille Russet, or that Red Eye Russet. For zebra chip, which was a big thing initially, we did lots of screening. Actually, the program, before I even came on board, tested more than 900 clones, most of them susceptible. With one of the students, Sam Viju, we ended up looking at tetraploid and diploid materials. We ended up, after screening like 3 rounds in different places, we ended up at the tetraploid level with 2 members of 2 families of the tetraploids that gave us tolerance. And one diploid clone that we are very excited about that is highly tolerant. I probably should not say resistant until I check and double check and triple check and so on, but we are very excited about it. Other avenues, chemical analysis to know and lead into health and nutrition aspects. We are looking at quite a number of things, and here I'm just showing you one example looking at amino acids. In particular, I want to point this first one that looks at methionine, and methionine ends up into a volatile that is giving the typical potato flavor. So in a way, this chemical will be associated with the potato flavor, and that Red Eye Russet seems to be doing pretty well. Antioxidants is another area of interest. And this graph represents 3 years of data of a subset of clones. And you can see on the left side is selections from the potato program, and on this right side is commercial varieties. So we have selections that have very high levels of antioxidants. Most of those are the ones with purple flesh or red flesh. Some areas for exploration in terms of phenotyping using unmanned aerial systems, and this is a collaboration with the group in Weslaco. This is the 2nd year, where we looked at yield and high temperature is a problem there, so indirectly looking at heat tolerance as well as zebra chip incidence. This experiment was not treated. And this is an example. You can see the growth of 2 clones. The one on the top is a Texas selection. There's the one on the bottom is Atlantic, which is a reference chipper. And in the pictures, we see for both of them canopy cover and excess green. And so we are now thinking about kind of connecting those things and see if there are some good correlations that we can find. Alternative phenotyping is a collaboration with Dimitri, looking at the use of Raman, especially offset Raman, for detecting and differentiating tubers that are healthy versus tubers that have zebra chip symptoms, as well as in collaboration with another student looking at the diversity of the materials we have and figure out if we can tell more about the chemical composition. And another aspect that I'm very interested in, and Kirti Rathore and Stephanie are working on, is related to gene editing, and they've been working on knocking the granular bound sucrose synthase gene. So in this case, ended up with events that have a very high level of amylopectin. And in this case would be for industrial applications. There are some other groups that are collaborating into actually moving towards like increased percentage of amylose, which would go towards the resistant starch and high fiber. So all of those applications related with health and nutrition. Many people involved, and I'm listing here a few, including everybody in my team and quite a number of different teams at Texas A&M, as well as pretty much I think I'm collaborating with everybody in the US, so definitely my colleagues with the Southwest program, Colorado, California, and then Western Regional, Idaho, Oregon, Washington. I also have to mention David Douches in Michigan, who's been running the genotyping for the SNPs. And then big thanks to all of the contributors, all of the funding sources. And I also have to reemphasize again how thankful I am for the royalties that return back to the program to support the program, and also to the growers that are providing all of that in-kind support to the program. And that's the end of the talk. Thank you.
Thank you, Dr. Isabel, for the impressive presentation. Now we have time for questions.
So when you're breeding for like resistance to certain pathogens, is there like a loss, like when you try to like pyramid multiple pathogens on, like you have PVY and then nematode resistance, like is there a lot, like a yield loss or something like alongside that?
Well, that's a really good question, and I'm not sure I'm ready to answer that yet, because I'm not lucky enough to have one of those clones that have the multiple genes together. But I assume that there is probably a cost to the plant. So if the plant has to invest in the fence, some of the other pathways will be reduced. So in particular, the main interest in potatoes is to translocate sugars and start generating starch. So those pathways will be involved. So I would say I'm likely thinking that that would be the case. But there's going to be variation.
Thank you.
This is an excellent presentation. You mentioned protein content, but you didn't mention what are you doing to increase it, what is the range, and what's your plan? Because it's one of the major food item, and if you can increase even 1 or 2% protein, that will go a long way. Thank you.
Good. So the average protein concentration in potato is 4%. And my first step is to actually figure out what kind of broad range I have for the material that I have in my breeding clone bank. So at this stage, I'm processing the samples. I think I should be able to tell you what is my variation maybe next time. But I hope there is enough variation that we can select for, or that we can claim as a special feature that we have materials with higher levels. And actually, I should let you know that yes, there is material with much higher level based on the screening that we have been doing for the materials in regional trials. So there is variation. There are some materials that are much higher, up to 8%, which is kind of double.
Thank you for your presentation. So you mentioned that nutrition, increasing nutrition, such as protein and the phytochemicals is a major goal, but Americans also eat most of their potatoes processed. So how do a lot of those nutritions hold up under processing? And are the gains you're making in breeding just going to be, boiled away in the oil vat? Thank you.
So there are a number of studies that look at the presence of some of those compounds when it's fresh and when it's processed, and some of the processing reduces the amount of beneficial nutrients. But if you start high, even there is a reduction, you still probably have an advantage of a benefit of that. I am also just hoping as well that the moment novel products are developed that are easier to handle and people will get used to them, maybe we start cutting down the percentage that we eat for French fries or chips. All of that is cool, it's good. It's just not something that we should eat every day. Once a week is fine, but then if we do it very frequently, it might not be as good.
What sort of varieties do you grow in Weslaco? Which ones do the best?
So the growers in the Weslaco area, many of them have contracts with Frito-Lay. So many of the varieties they grow are Frito-Lay varieties for the shipping market, and the yields are really low, but the industry needs something fresh when nobody else has it. So still, it's really important that our growers focus on producing those varieties. So I would say mainly chips, but they also are growing some other types.
Marie, I have a question. So is a higher percentage of the protein present in the skin region of the potato as opposed to the flesh of the tuber? Is the level of protein higher in the skin or not?
In the skin definitely has higher levels of minerals. But I would say in terms of protein, just because the flesh is a larger percent, that would be contributing more significantly towards the protein intake. Because in many cases, even the skin is not consumed. But definitely the skin has, the membranes, the phospholipids, proteins, so they have those high level of proteins. But most of it, they are measuring in the tuber percent.
Yeah, I'm always a little amused because when I have potato, I like to eat the skin. My wife doesn't. But for those of us who do like to eat the skin, an easy way of raising the protein level from our consumption would be to have smaller tubers, which have greater surface area per volume, right?
Yeah, and that's a really fast-growing market.
Right.
As part of my breeding program, I'm breeding for small potatoes. And for the small potatoes, what I really want is clones that give me a really large number of tubers that are small.
Right.
And you are right. Just because they are smaller, everything is kind of more concentrated. So the more nutrition.
Easier to cook on the grill too.
Much faster, yeah.
Thank you so much, Isabel. You know that in the system now there is a push to use food as medicine and to make it more nutritive. So I wonder if you have a focus or going to have a focus on increasing the amount of amylose in potatoes, and if that is that going to impact quality, cooking quality? What does it take to increase it from 20% to something like 30% or 35% amylose?
So I'm collaborating with a few groups that are interested in that particular aspect. So increasing the relative percentage of amylose so you have more fiber, lower glycemic index. Seems like it's a little more challenging than increasing relative proportion of amylopectin. But we're approaching that following two different avenues. So one avenue is a conventional breeding. There are some clones that have a higher level of higher concentration of amylose, and then also from the gene editing perspective as well. And I don't think we have looked, I'm not aware of studies looking at the cooking properties, but if you change the type of starch, it makes a lot of sense that the cooking properties change, probably even the perception in terms of texture. So lots of those studies need to be done.
I have a question. Hi, Dr. Vales. So in the varieties that you showed have a higher antioxidant levels, do they produce or yield at the same levels as the more popular varieties, or are they more aimed towards a specialty market?
At this point, the yields are lower than the top russets. There's just so much research and so much breeding that was done for the russets for processing and for fresh market that definitely the specialties have lower yields, but they could have much higher value. And so maybe that's kind of compensating.
Any more additional questions? Okay, let's thank Dr. Isabel for the presentation.